Good day to all. Today I am going to present my research finding on exploring Saccharomyces abundance in Sri Lankan grape microbiome for wine fermentation. These are the outline of my research presentation. Next, we have a brief introduction. The wine fermentation is a process carried out by the wine microbial consortium, specifically Saccharomyces, non-Saccharomyces, and bacteria. Generally, wine fermentation consists of two stages: alcoholic fermentation and lactic acid fermentation. Commonly, Saccharomyces cerevisiae and Saccharomyces bioanus are the major starter cultures used in wine industry for the production of wine, which results in consistency in quality and lacking of flavor complexity. Spontaneous fermentation is a complex and natural microbial process carried out by the wine microbial consortium that's naturally inhabiting in grapes. Generally, the final sensorial properties of the wine depend on several factors like grape varieties, geographical features, climatic condition of wine jars, and wine microbial consortium that present in the grapes. Among these, the isolation of the best wine microbial consortium from the grape skin, which can be used for the production of polyculture, is the crucial factor that determine the final quality of the wine. Up to now, there is no indigenous Sri Lankan wine starter culture used in local production of wine. So, efficient wine fermenting yeast five types from the Sri Lankan grape varieties have to be identified. These yeast types can be identified by studying their fermentation kinetics and their wine physicochemical characteristics. So, on the whole, by using those best strains, suggesting that it can be used to develop wine fermenting polyculture that will popularize Sri Lankan wine flavors all over the world. So here my objectives are to isolate the best wild type Saccharomyces yeast strain suitable for the wine fermentation from Sri Lankan grape varieties, and also to correlate their microbial fingerprints to their sensorial properties of the wine. Let me see the methodology in a brief format. First one is the sample collection. Here samples were collected from northern province, specifically Undra and Pali area, and Israel blue is the cultivar that they have isolated. Second one is the isolation of wine fermenting microbes. Here, isolation was carried out in two stages. First one is from the grape skin, and second one is from the fermenting must. And fermenting must, in the sense, the microbes are isolated from initial, middle, and later stages of fermentation. And isolated microorganisms were stored at specific parameters as mentioned here. And after that, monocultures were produced and microscopic observation was carried out for the yeast and bacterial identification. Later, biochemical analysis was carried out for the isolated microbial strain. After that, molecular analysis was carried out. Here, DNA extraction, PCR, microscopy electrophoresis was carried out, and DNA samples were sent to Microgen, South Korea for IDS sequencing. After the identification of yeast strains, wine samples were produced from the identified yeast strains, and their physicochemical analysis was carried out. Further, for the identified yeast strains, fermenting kinetics and the phylogenetic analysis also carried out. And in the right side of the slide, you can see these are the major tests that we are studying. Next, we move on to the results and discussion part. Here you can see the microscopic images for the isolated microbes. Here you can see the yeast cell with the buddings and rod-shaped bacteria, coca-shaped bacteria, fungal cells, and also the rod-shaped bacteria with chain structures also you can see. And here this table shows the fermenting ability and the biochemical analysis of the isolated yeast strains. Here you can see the both strains have positive results for phenotypic glucose growth medium and also in grape must medium. In the sense, they have positive sign for the color change and the carbon dioxide formation. Color change in the sense they have ability to produce the acid because ethanol and carbon dioxide are the main byproduct of fermentation. Hence, we can confirm these two strains have good ability of fermentation, and also they confirm the presence of budding, and also their pH and Brix also studied. And this the table shows the results of the molecular identification of the isolated yeast strains. Here you can see the both strains were identified as Saccharomyces cerevisiae and Saccharomyces boulardii. Both are best by fermenting yeast strains, where the Saccharomyces boulardii have some probiotic characteristics as well. And this diagram shows the yeast population dynamics and the fermentation kinetics of the identified yeast strains. Here you can see both Saccharomyces strains have better survival rate in alcoholic condition. And the blue line depicts the colony count. Here you can see there's a sudden drop in the colony count after 2-3 days of fermentation in the control samples. At the same time, both Saccharomyces strains, there's no any sudden drop as they have better survival ability in alcoholic conditions. At the same time, they have better sugar consuming ability also compared to control samples. And this table shows the wide fermenting kinetics of the identified Saccharomyces strains. Here you can see Saccharomyces strains, they have short half-life and better growth rate compared to Saccharomyces boulardii, because Saccharomyces cerevisiae consume the sugar effectively and grow quickly. At the same time, boulardii grow gradually in the alcoholic medium. And this diagram shows the FTIR spectrum for the wine sample produced by the respective identified G strains. Here you can see the major peaks that's present that represent the major compounds present in the wine sample. Accordingly, hydroxyl group, isotensine, alkynyl, phenols, tertiary alcohol, primary amines, ethers, disulfide halocompounds, and aliphatic compounds are the major compounds that present in the wine samples, and these are the major peaks with the respective wave numbers. And this radial diagram shows the ENOS results of the wine samples produced by the identified G strains. Then you can see Saccharomyces cerevisiae show higher sensorial values compared to other wine samples. At the same time, all three wine samples shows response to almost similar senses. On that way, R6 broad methane, then R7 sulfur organic, and broad alcohol, and broad range are the major sensors, so effective response for the wine sample. On that way, both Saccharomyces cerevisiae and Saccharomyces boulardii share almost similar sensorial properties, which can be confirmed by PC analysis as well. And yeah, this is the byplot of the PC analysis for the results that we have obtained from the ENOS test. Here you can see most of the sensors are shared between Saccharomyces cerevisiae and Saccharomyces boulardii. At the same time, the control samples was situated a bit far away from the other two samples. So we can confirm that both Saccharomyces cerevisiae and boulardii have almost similar sensorial properties. And this diagram shows phylogenetic analysis of the identified G strains with the other wine fermenting Saccharomyces strains available in other countries. Here you can see both Saccharomyces boulardii and Saccharomyces cerevisiae have different ancestors. Hence, we can use these both Saccharomyces strains for the polyculture making with the non-Saccharomyces or the bacteria, which results in novel sensorial properties in the wine. So these are the conclusions that I have arrived here. You can see Saccharomyces cerevisiae and Saccharomyces boulardii were isolated from the Sri Lankan external group cultivar, and Saccharomyces cerevisiae exhibited significantly higher rate and lower half-life, while Saccharomyces boulardii have extended half-life and low growth rate. And both strains have similar sensory qualities, slightly favoring the Saccharomyces cerevisiae. And we have to do the further sensorial and physicochemical analysis to find out the exact difference between these two strains. And with the distinct evolutionary traits, we can use these trying to make the polyculture that fostering innovations in the Sri Lankan wine industry. And finally, I acknowledge my principal supervisor and my co-investigator, Faculty of Graduate Studies, Department of Biosystem Technology, Faculty of Technology, University of Sri Lanka. And these are my references. And thank you.